Senescence Model. If you want to do research with the aged cells, how can you get them? For example, if you want to use mouse aged cells, should we just wait until the mouse naturally gets old? Let's check the lifespans of our representative mammals. Homo sapiens, humans, we can live 80, maybe 100 sometime soon. And monkeys' lifespan is usually 20 to 30 years. Laboratory mouse is 2 to 3 years. It's quite long. Now, master degree is usually 2 years. How long should we wait until you get aged cell from mouse? We can move the problem into a cellular level. If you grow the cells over and over, eventually the cells get senescent. And this is the model we call replicative senescence model. Alternatively, we can add oncogene into the cell, and it can generate the senescence symptom. This is called as oncogene-induced senescence. So the method is several different kinds of stress. Then the stress can induce premature senescence. To make a replicative model, we can grow the cells, and then when it reaches the confluent state, we can simply dilute the cells and then replate into the new plates. Then this process is called as a passaging, and from the passage 0, now we have a passage 1 cells. If this passage 1 cell grow and then gets confluent again, then we can dilute them further and make it into a passage 2. And this process can be repeated until the cells really can get senescent. When we grow the cells, in the beginning, the telomere has long enough the sequences and it can protect the chromosome. Now, during those replication steps, the telomere gets shortened and shortened. And by the experiment, the scientist Hayflick, he found that from human fibroblast, human cell can divide about maximum of 50 times. And the passage 50 cells has very shortened the telomere, and it cannot protect the chromosome anymore, and it cannot divide anymore. Oncogene also can induce senescence. Originally, oncogene is the gene which causes cancer development. These genes can be transferred by lentivirus or retrovirus. These are for long-term expression. And adenoviruses also can be used for transient expressions. And this virus can be used for delivery vectors. For example, when retrovirus infect target cell, the genomic information of a retrovirus is stored in single-strand RNA, and reverse transcriptase, which also carried by the virus, make complementary cDNA for the viral gene. Eventually, the double-strand gene is integrated into the host genome, and then those genes can be expressed by RNA polymerase, and eventually it makes the large amount of oncogenic proteins. This accumulation of cellular damages by the oncogene protein can induce the senescence. This kind of a viral infection can be monitored or modulated. So if this is the oncogene and then promoted here, and in the construct there can be markers such as a GFP protein, green fluorescence protein, can tell which cell is properly infected. Also, some of those inducible modules can be inserted. Then by adding the controlling compound, then we can control the time of effect. Stress also can induce premature senescence. Those stress include DNA damaging agent, or oxidative stress, or radiations. During the cell division, the DNA should be released from supercoiled state. Here the topoisomerase can release those stress. Also, if there is any errors in the DNA, it can fix it. So proper DNA duplication can be successfully achieved. But if we add the drugs, which can bind to DNA or bind to topoisomerase as inhibitor, then this topoisomerase cannot repair those damaged DNA, and eventually this damages is accumulated and it can generate senescence. So this is one kind of example of a chemotherapy-induced senescence. Also in the cell, mitochondria generate ROS, and then it can potentially make oxidative DNA damages. Mimicking this effect, if we add the oxidative stress from outside, then it can damage the DNA, and this is oxidative stress-induced model. How can we detect and discriminate the senescent cells? First of all, the morphology is changed. Senescent cells usually becoming much bigger than young cells, and the shape is quite heterogeneous. Also, the senescent cells express large amount of beta-galactosidase. The functional role of this enzyme to the senescence is not clearly identified, but this has been used as a surrogate the biomarker for senescence. So utilizing blue the product forming substrate, and beta-gal has been used for a long time to detect the senescent cells. The last, the senescent cells secrete a lot of the inflammation factors. This is called as a SASP, or senescence-associated secretion phenotype. So by detecting this secretion material, we define whether these cells are senescent or not.